understanding how endotherms adapt to the cold. And we know that both rodents and birds have this really a unique ability to adapt to tremendous shifts in temperature. And mice, for example, in the wintertime can adapt to the cold and maintain their core body temperature at 37 despite the ambient temperature being quite cold. And so how does this happen? How does metabolism change? And what are the key transcriptional pathways that drive this response? And so what I'll talk to you about today is primarily our focus on non-shivering thermogenesis. And non-shivering thermogenesis occurs in brown fat and beige fat. And brown fat, for example, is activated acutely with cold exposure, and through an uncoupling protein can drive heat production. And this can be activated within minutes of cold exposure. While beige fat, on the other hand, it's a thermogenic cell that also has this ability to uncouple, also has these metabolic programs that allow cells to drive futile cycles. But these cells appear after prolonged cold exposure. So this is more of a long-term adaptive mechanism. And what's pretty interesting is that we can essentially convert the subcutaneous white fat into beige fat with prolonged cold exposure. And there's quite a bit of evidence that this ultimately can lead to systemic changes in metabolism, improvements in glucose metabolism. White fat also has an important role in adaptive thermogenesis, as it provides a lot of the fuel that's necessary for animals to adapt to the cold. Now, humans also have brown fat, and it's cold inducible as well. And this has been highlighted by many different groups now, showing that using glucose tracers, showing that there's an increase in glucose uptake in brown fat cells in response to the cold. And you can see these regions really light up with the glucose tracer. And it turns out that these are thermogenic cells that express the uncoupling protein that have been largely defined as beige fat and brown fat cells. And there's also this inverse correlation between brown fat activity and percent body fat. So individuals that are lean tend to have greater levels of brown fat activity. So the amount of brown fat that you have may ultimately impact systemic energy metabolism. And we know certainly in rodents that when you change the ambient temperature, you can really dramatically change energy expenditure. So these are some experiments that we did using the CLAM system to monitor energy expenditure and food intake. And what you can see here is that when you place mice at 30 degrees and you compare that to 16, you see a doubling in energy expenditure. And this is primarily non-shivering thermogenesis. And as we lower the temperature, we also start seeing effects from shivering as well. And during this shift of lowering temperature, we see that the RERs decrease, which suggests that there's a shift towards utilizing more lipids. And so metabolically, the mouse is shifting its program towards using primarily lipids as a source of energy. And so based on this observation, we've decided to look more closely at what are the lipid changes that occur in response to the cold. And the approach that we've taken is to use untargeted LC-MS analysis of plasma lipids and begin to define what are the broad changes in lipid metabolism that are occurring. And this has really led us to interesting biology and physiology that we're continuing to try to understand. And so this is a result from the lipid analysis. Here's a heat map showing changes in the plasma lipids. And you can see that there are about 281 lipid features that change. A third of these increase in response to the cold. And when we look more closely at which lipids are changing through this volcano plot, you can see that there are a number of long-chain acylcarnitines. And long-chain acylcarnitines essentially have an acyl chain in one end and a carnitine group on the other. And the carnitine essentially allows these lipids to become soluble in aqueous solution. And so what was intriguing is that these were primarily long-chain acylcarnitines. And when we looked more closely at several acylcarnitine species, we saw that the shorter chain acylcarnitines weren't changing as much as the long chain. And so this was surprising in some ways because from what we know from what's been published for several decades now, is that once you generate acylcarnitines, that these lipids are destined for fat oxidation. And what you can see here is the pathway that's involved in acylcarnitine metabolism, with the primary enzyme CPT1, which takes a fatty acyl-CoA to make acylcarnitines, and then these acylcarnitines can be transported into the mitochondria and oxidized. But we were seeing these in the plasma. So what was driving this change? So there were several questions that we had. Where were these acylcarnitines coming from? What was their role in thermogenesis? And what is driving the transcriptional regulation of this pathway? And so when we looked at several different tissues, we found that one of the tissues where there were a number of changes in some of the enzymes and transporters involved in acylcarnitine metabolism was the liver. And we saw both CPT1A increase by mRNA, and we saw also, CPT1A increase by Western blot analysis, and so did CPT1B. And so this really focused our attention to trying to understand how the liver changes its metabolic program during the cold. And at the time, there was very little known about what the liver's role is in adaptive thermogenesis. And so we were really excited to delve into this aspect of metabolism to try to understand what the liver was doing. And so the next experiment we did is to target this pathway by knocking down CPT1A and CPT1B. Now, CPT1A is the major liver isoform. And when we knock down CPT1A, we can see dramatic changes in long-chain acylcarnitine species. So here, relative to the scramble control, you can see that CPT1A knockdown lowers palmitoylcarnitine. And when you knock down CPT1B, the effect isn't as dramatic. Now, when we knock both of them down, we can see that the effect is quite significant. So then we put the mice in the cold to test what is the role of this pathway in driving thermogenesis. And keep in mind here, we're only targeting the liver. And as a result, what you can see here is relative to this control, which mice normally do quite well in response to acute cold exposure. You can see that when we knock both of these enzymes down, we can see cold sensitivity. So the core body temperature dropped quite dramatically. Now, to address whether it's actually palmitoylcarnitine or an acylcarnitine species that's driving this response, we reintroduced palmitoylcarnitine to the knockdown group. And you can see that when we do that, we can rescue some of the cold sensitivity that we see in the knockdown experiments. And so this really highlighted to us the importance of driving acylcarnitine production, but also it highlighted the role of the liver in contributing to the plasma acylcarnitine levels, but also contributing to adaptive thermogenesis as well. We next asked, where do these acylcarnitines go once they're in circulation? And so for this experiment, what we did is we did a tracer study where we used radio-labeled palmitoylcarnitine, and we placed mice in the cold room, and we followed we followed the radio label in the blood, and you can see that within an hour in the cold, almost 75% of that label has been depleted versus at room temperature, you see that only about 20% has been removed from circulation. So it indicates that at least with cold exposure, there's a high clearance rate in response to the cold. And then when we looked at several different tissues to see where the radio label was ending up, we saw that there was a significant amount that was ending up in the brown fat. So you can see relative to room temperature, there was an increase in response to the cold. Notably, there were other tissues that increased acylcarnitine uptake, including the heart and the skeletal muscle. In contrast, the white adipose tissue in the liver had reduced uptake of the radio label, which in some sense for the liver, it makes a lot of sense if the liver is generating these acylcarnitines. It doesn't make a lot of sense for the liver to also take these up in response to the cold. And so this led to this model of how the liver is involved in adaptive thermogenesis. One of the things that came out of this study is that the adipose tissue is also an important driver of the thermogenic response, and primarily by providing fuel for thermogenesis. And so to address the role of the adipose tissue and free fatty acids in driving the changes in the liver, we decided to look more closely at this pathway and targeting one of the enzymes that's involved in lipolysis. And so the experiment was very simple. We essentially utilized ATGL. Sorry, guys. Oh, thank you. That was my son with some coffee. And so we looked at, we targeted ATGL in the adipose tissue by crossing ATGL flox mice with the adiponectin Crete. And one of the major questions that we had is, what is the role of lipolysis in driving the changes in the liver? And this also led to deeper questions of what are the lipids that are being secreted by adipocytes in conditions like thermogenesis, and whether these lipids could also be driving the transcriptional changes in the liver. So this system is really nice because it allows us to essentially block lipolysis and then look at the impact on the liver. So here's a Western blot just showing that we have an ATGL knockout in the adipose tissue. And we also see that free fatty acids are decreasing in these knockout mice, and that in response to beta-3 adrenergic receptor agonists, which from now on I'll just call CL, essentially it stimulates lipolysis in the white fat and increases free fatty acid release. Then to address whether the changes that we were seeing in the liver were due to lipolysis in the fat, we looked at expression of a number of the enzymes involved in acylcarnitine metabolism, and as well as other enzymes, other pathways that we identified changing in response to the CL agonist. And you can see that in the liver, we can see an increase in CPT1B and OCTN2. However, when we knock out ATGL in fat cells, we no longer see that response. Now, keep in mind that the CL in this case, it's pretty selective for fat, both white fat and brown fat. And there's no receptor in the liver. And we've also used this drug on hepatocytes in vitro, and we don't see this response. So this is definitely highlights that this is communication from the adipose tissue that's occurring to the liver. Notably, when you knock out ATGL in the adipose tissue, it really dramatically impacts lipid metabolism in the liver. And we've done some lipidomic work now looking at a number of lipid species in the liver. And one of the major lipid classes that changes dramatically in response to CL is triacylglycerol. And you can see relative to the control here, CL can increase triacylglycerol quite dramatically in the liver. However, when you knock out ATGL in fat cells, there's a dramatic reduction in these knockout mice, and you no longer see the response with CL. And so this highlights, one is that the adipose tissue is adipose-derived lipids are a major driver of the lipids in the liver. And that for conditions like hepatic steatosis, for example, this may be a significant pathway that should be targeted to prevent conditions like NAFLD and NASH. We've also taken the livers now and using a similar paradigm to look at broad transcriptional changes. And when we do pathway analysis, we can see that there are a number of pathways involved in lipid metabolism, which is what we would expect. But surprisingly, that there are also pathways that are being affected involved in carbohydrate metabolism, purine and ribose and nucleotide metabolism. And so there's quite a bit of work, I think, that we need to do to now understand how adipose tissue lipolysis, in addition to affecting lipid metabolism, is affecting other aspects of metabolism in the liver. Now, taking the data set from mice that were treated with CL, we can use this tool that's developed by the Ingenuity Pathway Analysis, where we can essentially predict upstream regulators of these transcriptional responses. And when we do this, what we find is that PPAR alpha is one of the top putative upstream regulatory pathways of this response, which is in a lot of cases not too surprising based on what we know about PPAR alpha and its role in lipid sensing in the liver. But what's also interesting is that we see a number of other pathways that have not been necessarily linked to fatty acid sensing or an influx of fatty acids from the adipose tissue. Some interesting upstream regulators include P53, the glucocorticoid receptor, SREBP, 1C, and we also see that there is activation of the ER stress response, which makes some sense as there's an influx in fatty acids, and these fatty acids ultimately end up in the ER and have to be assembled into triglycerides. And notably, we also see that there are changes in pathways that are regulated by HNF4 alpha, which we ultimately also showed is essential in this cold adaptive response in the liver. And we also did the same experiment in the ATGL knockout mice, where we've knocked it out in fat cells. And you can see that the upstream regulators that come out are not as significant as the control mice. And we see that a number of the upstream regulators that we identified now drop out. And so this has been a really nice system for us to really try to hone in on some of the key pathways that are important in lipid sensing in the liver and how the adipose tissue is driving that response. And just to sort of give you a broad overview, we can also look at some of the downstream targets of these pathways and look more closely at how they respond to CL. So for example, here we have vehicle on the left in this heat map, CL treatment. You can see that there are a number of PPR alpha targets that increase. And in the ATGL knockout mice, you see that these transcripts are no longer induced. And so it's also allowed us to find direct targets of these pathways that we can follow up to see whether they're important contributors of lipid metabolism in the liver. And again, we can take these data sets to be able to focus on the key pathways that are important in response to CL. So now, based on this observation, this really highlighted that there are signals coming from the adipocyte that are communicating to the hepatocyte. And so to look at this more closely in an in vitro system that is less complicated, since, you know, when you put these mice in the cold or when you give them CL, there's quite a bit going on in multiple tissues. And so to see whether we could recapitulate this in vitro, we decided to differentiate adipocytes. And once they were differentiated in vitro, then we treated them with vehicle or CL. Then we took that conditioned media and we placed that conditioned media on hepatocytes. Then we can follow up with changes in gene expression and also look at microscopy to see whether there were changes in lipid droplets and some other organelles. And so here's some immunofluorescence from an experiment where we used HEPA cell line. And you can see here we've used Bodipi, which will stain for the lipid droplets. And as you can see, relative to the vehicle group, the CL treated, the CL conditioned media really drove an increase in the amount of lipid droplets. And this occurs in, within a couple of hours after placing the conditioned media with hepatocytes. And the conditioned media was also able to drive some of the changes that we were seeing in vivo, where we had seen an increase in PDK4, which is involved in regulating fat oxidation. And we can also see an increase in CPT1A, which is involved in acylcarnitine synthesis and involved in fat oxidation as well. And so the next question we had is, what is in the conditioned media? And so we went back to using our untargeted approach to begin to define the lipids that were being secreted in the media, and particularly in response to the beta-3 adrenergic receptor agonist. And to our surprise, we saw quite a bit of lipids that were being released into the media. We found 791 lipids. And of these, there were 381 that significantly changed in response to CL. When we look more closely at which lipids are changing, here we just graphed the number of lipids within each major lipid class. And you can see that fatty acids several different fatty acid species are changing in response to CL, and they're being upregulated. And we also find that there were a number of triglyceride species that were decreasing in the media. And to some extent, this was kind of surprising to us, as triglycerides haven't been really characterized well as being released from the fat cells. So this was quite surprising. And we also see that there are changes in phosphatidylcholine, as well as ceramides, and some of the signaling lipids that have been published more recently, like FAFAs. There were a few that were upregulated in response to CL. And so based on this initial observation, we then did a targeted approach and looked more closely at the abundance of these lipids. And as we would expect, when we give CL to adipocytes, we see that there's quite a bit of free fatty acids that end up in the media. And the major fatty acid species that we see are actually palmitates and palmitoylate, and some stearate and linoleate. We can also see that lysophosphatidylcholine increases, and there's smaller effects on lysophosphatidylethanolamine and phosphatidylethylamine. And as you can see, we also found triglycerides in the media as well, and that seemed to decrease in response to the beta-3 adrenergic receptor agonist. And so in summary, this is what we've discovered based on our studies and understanding how cold exposure can change metabolism, and that the adipose tissue is a major driver of hepatic lipids, particularly triglycerides, and can also drive changes in gene expression. And we found that HNF4 alpha is a major driver of this response. But also that these free fatty acids can provide the substrate for generating acylcarnitines. They can also provide substrate for generating ketones, which ketones also increase in response to the cold. And so again, the liver in this case is providing the essential fuel that can be utilized by peripheral tissues like brown fat to drive heat production and cold adaptation. And so in conclusion, cold exposure promotes energy expenditure, lipid utilization in the liver, and changes in the plasma lipidome. Long-chain acylcarnitines are elevated in response to the cold. And these acylcarnitines can be taken up by brown fat with cold exposure, in addition to the heart and skeletal muscle. We've noted that the adipose tissue lipolysis governs the transcriptional response in the liver, and is a major contributor to hepatic steatosis. And there are pathways involved in fatty acid oxidation and lipid droplet biogenesis that are upregulated in the livers and in hepatocytes when we activate lipolysis. And adipocytes are releasing multiple lipids in addition to free fatty acids. And we think that these lipids may be important drivers of transcriptional changes in the liver, may also be important in communicating to other cell types outside of the adipose tissue. And notably, these adipocyte-derived lipids can profoundly impact hepatic gene expression, both in vivo and in vitro. So I'd like to thank the people that did this work. This work was initially started by really talented postdoc, Judy Simcox, who now has her own lab at University of Wisconsin. This work has now been carried out by Xixiang Zhang, a really talented graduate student in my lab at UCLA. And I'd like to also thank other personnel in the lab, graduate students and postdocs that have been a really important contributors to our study and cold adaptation. We've also have collaborated with a number of investigators and both at UCLA and at the University of Utah. I'd like to thank Aaron Kershaw and Rudy Zechner and Renat Schreiber for their help in some of the studies that have been completed with the ATGL knockout mice, and certainly the lipidomics core at UCLA. and as well as some of our more recent work with some of our collaborators at UCLA, like Tamer Salem and Zhengyi Zhang. So with that said, I'd like to thank you all for your attention, and I'll open up for questions now.
Hey, excellent, Claudio. This was really good. So again, everyone, if you have any questions, please use the raise your hand function in the chat, I think. And then, well, while we're waiting for questions, I would like to start with the first question, Claudio. So, since you're seeing these, this cold-induced changes in adipose tissue lipolysis impact the hepatocyte acylcarnitine metabolism regulating genes, and then the downstream effects, I'm wondering if you see the same liver response during conditions of fasting or feeding or insulin injection, like during normal physiology, where adipocyte lipolysis changes, other than cold-induced changes?
Yeah, so we've primarily focused on cold exposure. I guess the other case where we do see an increase in acylcarnitines is with the CL drug, where we're directly stimulating lipolysis, right? Mm-hmm. In other conditions like high-fat diet, for example, that's been shown to lead to elevation of acylcarnitines. And they've certainly been linked to insulin resistance, but it hasn't been entirely clear why. And one possibility of why they're increased in models of insulin resistance may be due to the impaired suppression of lipolysis. So in conditions of obesity, you tend to have a higher free fatty acid levels, and that ultimately may be driving some of these increases in circulating acylcarnitines.
Mm-hmm. Okay. Yeah, because you also mentioned that ketones, they are converted to ketones, right? So that makes sense if this pathway also gets activated during fasting, But okay, this was excellent. So we have two questions. One of them is Zhu Wei Zhu. Sorry.
Oh, no, you're fine. So very nice talk. So I have a question is, because you showed that the liver-derived acylcarnitine the white fat tissue actually have decreased uptake for the liver-derived acylcarnitine, but the brown fat increased. So I just wonder whether it's because the acylcarnitine receptor is quite different in these two different types of the fat. The secondly, because in your introduction, you show the white fat can also, become brown fat like the beiging, so during the cold exposure. So I just wonder, for the brown fat, the difference between the brown fat and white fat in terms of the fatty acids oxidation. So what is the difference?
Yeah, great question. So I think that's a really nice observation, right? That these acylcarnitines are going to brown fat, but you see the complete opposite response in white fat. So we haven't looked at beige fat. So when we do these studies, usually mice are at room temperature, and then we place them in the cold for 5 hours. And so we're looking at acute cold exposure. And so in that context, we normally wouldn't see the inguinal white fat brown, for example. So you wouldn't see beige fat cells in this time window. But I think it's quite possible that if we did a prolonged cold exposure, we had lots of beige fat cells in the subcutaneous fat, that you might see more uptake.
Right. I'm also wondering. Oh, sorry.
Sorry. And then what's driving the difference between these two? We really don't know. If the transporter that's involved in taking up acylcarnitines hasn't been identified. And so that's still an active area of research.
Okay. Yeah, I'm just wondering, for the, if you do the long-term co-exposure, so we assuming probably there's increased lipid oxidation in the white fat. So I just wonder why it's because there's more endogenous fatty acids. So those can convert to carnitine for the lipid oxidation. Instead, they use the liver-derived carnitine. So that is what I'm wondering.
Right. So let me just try to understand your question. So you're wondering why acylcarnitines would be needed when the brown fat can make its own acylcarnitines?
No, I'm wondering what probably is the difference between the white fat and brown fat is because the white fat doesn't need the liver-derived acylcarnitine because itself already can make acylcarnitine.
Oh, I see. Yeah. So I think the white fat is probably not really making much acylcarnitines because you need mitochondria to do that, right? And so white fat is going to have very few mitochondria. And so, one possibility is could be a flux issue. Since brown fat has a ton of mitochondria, it can act as a sink for these acylcarnitines, and white fat doesn't necessarily have that capacity, right? Because white fat has very few mitochondria.
I see. Okay, thank you.
Great question.
Okay, and then we have another question from Alex.
Dear Claudio, this was a very interesting talk, and I'm glad that both of our talks will have a lot of lipids. That's great. My question is directed towards the ATGL knockout mice. As you know, these mice are extremely insulin sensitive, probably because of the lack of fatty acid flux to beta cells. Have you tested whether some of the responses you're not seeing is because they're just hypersensitive to insulin? For example, if you give more insulin, you can rescue some of these effects, or is it really only the lipids?
Oh, great question. So we haven't really looked at the role of the insulin, or the insulin signaling pathway in this response. But we can certainly recapitulate some of this response in vitro, right? So if we treat the adipocytes and take the conditioned media, we can see a lot of the changes in lipid metabolism in vitro that we're seeing in the liver. And this is taking insulin out of the picture. And so I kind of doubt that insulin would play a role, at least in some of the transcriptional effects that we're seeing. But it's possible that we're missing some of this in vitro, and it's an aspect that we need to really explore more. Because certainly, there has been some work, right, showing that insulin is driving some of the changes in brown fat, and that seems to be important in driving lipid uptake.
Thank you.
Okay, Ebru has a question.
Yeah, I can't raise my hand, so this was a great talk, Claudia, because it made me really appreciate how collaborative a response this is to cold-induced thermogenesis. So it's not just brown fat, but a coordination of things. I mean, you would expect that from life anyways, right? A lot of collaboration between the organs. So it really made me appreciate that. And I have a question. So in obesity, when the adipose tissue and liver converts to a lot of de novo lipogenesis and activation of SCD and generation of monounsaturated fatty acids, how does this response change between the three organs that you're talking about? Have you looked into that?
How does desaturation change?
How does desaturation and the production of monounsaturates by liver and adipose tissue play into effect?
Great question.
Is SCD important in liver and adipose tissue in that case? And does that change the thermogenesis response?
Yeah, I feel like I saw that SCD1 was increasing in response to the CL drug in the liver. And it came up as one of the targets of SREBP1, right? Certainly fatty acid synthase was up as well, and those two tend to be up together often, which in some cases is kind of puzzling as well, right? Because there are fatty acids that are coming from the fat tissue, yet the liver also seems to be trying to turn on this transcriptional response to drive fat synthesis. But we haven't directly measured fat synthesis yet, so I don't know whether the transcriptional response really matches the actual metabolism in the liver. But there seems to be some evidence that SCD1 does go up in the liver. We haven't looked at other tissues like the brown fat or white fat, and I don't recall whether that changes in that context.
And one of its major products being palmitoylic acid, I think I saw that in your screen coming up as an increased fatty acid in your liver.
Yeah, it increases when you activate lipolysis, and so that's coming from the fat.
Yeah. So more of that might come out of the fats and the liver in an obese animal, for example.
Yeah, you would see, you'd likely see more of these circulating in the blood in a high-fat diet.
Mm-hmm. Thank you.
Yeah,
Ebru, I can maybe also comment on this. So we've done some stuff with the lipid flux there, and it's clearly that somewhat paradoxically, despite the fact that cold is in catabolic state, there is increased de novo lipogenesis in brown fat and beige fat. And some of this might actually be related to the high glucose influx, the activation of CHRBP, and the concomitant increase in de novo lipogenesis. It might very well be that the tissues need some specific de novo lipogenesis derived fatty acids, which they otherwise don't get. And there's clearly a crosstalk between liver and adipose tissue in this context.
Thank you.
Okay. Do you think we have a few more minutes for one more question, or it's 12:48? Do you think we should switch to the other talk? What do you think, Ebru?
Just a quick question, maybe. Oh, I guess
Okay.
that person decided.
All right.
Okay.
All right. Thank you, Claudio. It was excellent. Thanks again. And then now Ebru will introduce Alex.
Okay. Yeah. So thank you to Claudia for a great talk and discussion. And so we're lucky to have both Claudia and Alex present today because their work complements each other, I think. And so Alex and I did our postdocs in the same lab, but you know, we work on a little bit of different fields. Alex worked with George Harine and with Gökhan Otomishtegil, and he's really been working on brown fat for a long, long time. Now he has his lab at LMU, a great place to do research. So I'm really looking forward to what he has been able to accomplish since he moved there. And what he's going to accomplish is going to be great. I'm pretty sure of that. So go ahead, Alex.
Well, thanks for the nice introduction, Ebru. I do have to apologize, though. My talk will be a little bit more conceptual because I wanted to merge the concepts of cardiovascular disease and obesity. And you might then realize at the end of the talk that these two large, broad topics hardly actually in our basic research talk to each other. And there's a big need that, and particularly for understanding cardiometabolic disease, we provide some more, interaction between these two topics. So if you really want to learn about the latest shit that's coming out of our lab, I have to say maybe this is going to be the next round of the virtual seminar series. However, I would like to dedicate this talk also to a colleague of mine who recently passed away, Jimmy Berbe from the Netherlands. Most of the data I will be presenting today, he was involved with. We even have a co-first author paper together, and his untimely passing at the age of 42 is really saddening. It was just this May, and I want to dedicate this presentation to his legacy. And so why did I say that cardiovascular disease and obesity are very important? If you look at the cardiac mortality in the 20th century and in the decades to come, what you can see here is that at the beginning of the century, towards the 1950s, because there was a decline in infectious diseases, better food supply, better transportation, the cardiac mortality went up. And of course, this is shown for the US, but it's relatively representative of the Western world. Then after 1950s, 60s, the cardiac death rates went down again because we had better medical and surgical procedures, better public health strategies, and smoking has been pretty much banned for our public life. Now, what is happening now that we see a surge in overweight and obesity that is having an epidemic state right now, and it is predicted that because of the childhood obesity we're seeing now, a along with the rates in obesity, we will see a cardiac death and mortality rates like we have never seen before in the next decades to come. And why is that? The overweight and obesity, the increased adiposity, too much fat, has a variety of mechanisms by which it impacts, at the end of the day, cardiovascular health. And this is through hormonal regulation, adipokines, cytokines, and insulin. And one manifestation here is type 2 diabetes. There is the concept of too many lipids and lipotoxicity, which in the end can lead to NASH and cirrhosis. Dyslipidemia, which I will be talking about a lot, then fosters atherosclerosis and coronary artery disease. And of course, there are also other concomitant factors that are bad for you. So energy balance at the end of the day is a matter of energy intake and energy expenditure. And in a simplified concept, of course, it's what you eat and what you drink. And we know that our processed foods and sweetened beverage are really bad for our carbohydrate insulin relationship. And those calories then also are not metabolized equally and determine our energy intake. On the other hand, we work when we expend energy, and this is done by our body, the basic metabolic rate, our thinking, some of the thermogenesis we've already heard about, and exercise, obviously. So this is a really important process, very simple concept, but really at the heart of our obesity problem. And how now do we study this? And I think this is where I think it gets interesting, because you can see this is a very nice picture I took from this recent review, where you can really look into a model system with high throughput and mechanism to a difference in relevant to human physiology. And you can see that the non-human primate models, of course, are very close, but it's very difficult to work with. And our rodent models, obviously, are great, but they are, like it is said here, not really representative of most human diseases, and particularly not in terms of cardiovascular disease. And if you think about it, how we then actually measure energy metabolism and particularly also brown fat function, these days there are many, many factors that impact on how we study this subject. And you can see here that it sounds easy to put mice on a high-fat diet and see how do they get obese. But there are so many factors, actually. It is not a simple science. It's involved by the genetic background, where you get your animals from. I don't want to say the word gut microbiome, but I think I have to. Which diets, where did you get those from? And of course, are you looking at males, females, and is your animal caretaker maybe a little bit too loud once in a while? And then also I want to mention here, there is this concept of metabolic cages, indirect calorimetry. This is a very old school system. And I brought this picture up because maybe Ebru, you remember, this is in Harvard School of Public Health. This is a 20-year-old machine. It's still running and it still gives great data. So of course, this is one of the gold standards, how we look at energy metabolism. And most people, you know, they find, let's say, a lean and an obese mouse. And when they measure the energy expenditure, machine sometimes gives you some values here that are expressed as kilocalories per kilogram. And then obviously the obese mouse, because it's so big, will always have less energy expenditure. However, then looking at the same mouse, just per mouse, the whole mouse, then sometimes there's no difference. And then the question is really, is it different, yes or no? And I want to stress here that there was recently a great overview that really picks up where this paper here in 2012 has left us. This is a review from Matthias Chupp and including Eric Ravison and many others that are really let's say the leaders of energy metabolism for the last 30 years. And what they've shown here is that you can take whatever random mice and just take these, let's say, averages, and you might find a difference where there is actually none. The energy expenditure is the same. And even when you have, let's say, two groups that are separate in energy expenditure, you can also, by choosing the wrong mice, end up not seeing any difference. So the take-home message from this is, and this is now the paper I want to mention here, it's Alex Banks and an international collaboration where they looked into many, many factors that determine energy metabolism. And you can see each individual dot is one mouse. And even though we know that males and females have a different energy expenditure in absolute values, they're not innately different in the way they burn calories. And I think it's worthwhile reading this paper. It tells us a lot. Now, energy metabolism, of course, is mainly influenced in mice by basic metabolic rate and temperature. We heard about the great contribution of brown fat. And this is the Harvard Medical School quad during a typical New England winter. It's very cold and very snowy, as you can tell, and that's why we eat brown fat for. So brown fat, we've heard, is neither fat nor flesh, is an interesting type in between, was discovered by the Swiss anatomist Konrad Gessner. And compared to white fat, these cells are small, have high mitochondrial content, look like raspberries, and there's an adipose tissue browning, which, as you all know, is the intermediate phenotype. So one of the early questions that was really not answered in the literature was, how does the fuels get to the brown fat? And I just want to spend some time here. You probably all know the Bible of brown fat, the 70 pages review by Barbara Cannon and Jan Nedergaard. But on this chapter, how does the fuel get to the brown fat? There's actually only maybe, let's say, 10 sentences or so. So when I was a young PhD student, we ventured into this topic and then was studying lipoproteins. And we've heard something about energy substrates already, but lipoproteins, triglycerides, just by mere numbers, are definitely the greatest, let's say, energy supply for most tissues. And there are different sorts of lipoproteins which carry sometimes more triglycerides, like chylomicrons and VLDL, and sometimes carry more cholesterol, like LDL and HDL. I will get to these classes later in more detail. And lipoprotein metabolism is a very interesting and very complex inter-tissue process, and it can be largely categorized in an exogenous and an endogenous path, where here you take up the lipids from the intestine. They are built into chylomicrons, which then deliver the fatty acids into the adipocytes, for example. And then the smaller remnant particles are cleared. And this is during feeding. And then in fasting, these VLDL particles are released from the liver, which then during fasting provide fatty acids, and then are transformed into LDL and cleared from the liver again. So you can see this picture here is from 2009, and there is just one adipocyte, a white adipocyte, and that was pretty much the picture 10 years ago, that brown fat was not really on the radar for, let's say, systemic lipid metabolism. So one of the things we were doing was actually just do a postprandial oral fat tolerance test. At that time, you give mice triglycerides, they go up and they go down. But when the mice were exposed to cold, those triglycerides hardly went up. And you can see when you separate and look into the plasma lipoprotein profile, that there are hardly any lipoproteins found in the plasma. So wherever these lipoprotein went at that time, it was very remarkable and a striking effect. So we wanted to study this process in more detail, and we developed an imaging platform where we incorporated these fluorescent nanocrystals into the core of these recombinant lipoproteins. And these are great imaging tools to, like a Trojan horse, track lipoproteins in the body. And they look like milky emulsions under visible light, but under UV excitations, they give you fluorescent colors depending on the size of the Q dot core. And one of the things we did was we really looked into the brown fat and looked at lipoprotein uptake in real time in a live mouse that was cold exposed and placed on a microscope. And you can see here that this movement is the breathing of the mouse. These black dots are erythrocytes. The blood is stained in red. Blue are the nuclei. And then the lipoproteins, immediately after the injection, they find the brown fat. And for example, you can see here within a minute or two, the complete endothelial layer of the brown fats are lined with lipoproteins, emphasizing that the brown fat is really powerful in sequestering lipoproteins from the plasma. And you can also quantify that by radioactive tracers, where you see that there is a roughly a 5 to 10-fold increase in lipid uptake by the brown fat. And then what you can also do is you take a mouse model that has pretty much cream instead of blood, and then you put those mice into the fridge. Overnight, they are cured. Now, this is all great. We did some other mechanistic stuff. So at the end of the day, we had some insight into lipoprotein processes in the brown fat actually takes place. And the obvious question with such a strong effect on plasma lipid homeostasis was obviously, how does this now impact the development of atherosclerosis? So atherosclerosis, as you know, is a double-edged sword where you get retention of cholesterol-rich lipoprotein particles, and then in a decade spanning process, you get these very complex atherosclerotic plaques, which then can rupture and can lead to myocardial infarction and stroke. And the pioneers of this field were Nikolai Anichkov, who was the first to propose that actually there is no atherosclerosis without cholesterol. And then Russell Ross was the one who coined the term that actually atherosclerosis and inflammation, an inflammatory response to insulin. So this is really was one of the founders of the chronic metabolic inflammatory field. But studying this in mice is a little difficult. Mice don't get myocardial infarction, and mice have very different lipid metabolism compared to humans. For example, mice carry all their cholesterol in HDL particles. This is why they don't really get atherosclerosis. And in men, you see that we pretty much have a 2-to-1, 1-to-1 ratio of LDL and HDL cholesterol. However, in the last decades, we have developed tools to make mice more human. And one way to do that is to take away the LDL receptor, which is usually the protein that clears LDL. And in a heterozygote, you get a little bit of LDL, and in a knockout, you get a large LDL peak, making these mice have a more humanized lipoprotein profile. And then there's also the dietary component, which is very important. So on chow diet, it pretty much looks like this. But when you give a cholesterol high-fat diet to these mice, wild types, they only get more HDL. But the LDL receptor knockout mice, they display a severe hyperlipidemia and then only develop atherosclerosis. So we thought, wow, that's a cool model. Let's study this and see whether when we activate the brown fat, can we actually prevent atherosclerosis? And we got very excited. That was even before the other paper was published, and asked the question, how does increased energy expenditure actually impact atherosclerosis development? And I remember we did this study in ApoE and LDL receptor knockout mice. As I said, these are the most frequently used atherosclerosis models. We had them in the lab. It was very easy, and we were very experienced with them. And then we did the study, and I took these blood samples out of the centrifuge, and I got, oh my God, it worked. But then I clearly looked at the labeling and then find out, well, okay, so the control mouse actually has less lipids in the blood than the cold exposed mouse. And that was very surprising and very confusing, because the hypothesis was that it should have been the other way around. And while we were doing this, a little bit later, a paper came out in Cell Metabolism where they were actually showing that cold exposure is bad for you. Cold exposure promotes atherosclerosis plaque growth. And we were like, yeah, well, we kind of see the same thing. But there are a couple of things that I think one needs to take into consideration. And one is that cold increases food intake. And I've emphasized that how important dietary cholesterol is for these models. And then we should also not forget that these models are artificial in the sense that they do not have a normal lipoprotein metabolism. And what we wanted to do is circumvent the cold, circumvent the effect on food intake. And what we did was use a model with beta-3 adrenergic activation. This can be CL, this can be Myabragon, which also works in humans in this study by Aaron Cypus, and did a pair feeding study, which means that we have a control group where we measure the food intake, and then there is an experimental group which gets the amounts of the controls. And using this regimen, then we were feeding the mice with a diet that contained the CL compound to activate the brown fat, and we matched the dietary cholesterol intake. So the mice ate the same amount of cholesterol, but they still had brown fat activation. And interestingly, then in this model, of course, we saw that the TGs were down, the brown fat was active, but there was zero effect on cholesterol, neither in the ApoE nor on the LDL receptor. So total cholesterol was sky high, but not higher and then the atherosclerosis was similar. And of course, this was disappointing. We thought like, ah, damn it, still brown fat seems to be not really beneficial. So we turned to our colleague Patrick and Jimmy, who I mentioned in the beginning, who passed away. And they were saying, well, you know, the Apoe and LDL receptor knockout mice, they are so messed up because these are integral and very important molecules. Why don't you try our model that is more human and is less severe? It's called the Apoe3 Leiden CETP mouse model. And there you have a human mutation. It's a knock-in mouse that only slows down the clearance, but it's still possible to activate this pathway with regular anti-atherosclerotic compounds. So this mouse model is actually not so severe, and it is susceptible to brown fat activation, because here in this model, both cold and Cl compound lower plasma lipids actually despite an increased food intake. And then obviously what we could also show that when you have the right mouse model, then brown fat actually lowers blood lipids and it also prevents atherosclerosis. However, the people that have published the first brown fat paper still keep continuing to push forward the idea that actually brown fat is bad for you. This was a paper from last year in PNAS where they show that myabragon again exacerbates atherosclerosis. We also did a study on myabragon again in our CETP line model, and we sent a brief report to PNAS with the data showing that, you know, this statement is kind of misleading. It's just the mouse model that prevents you from seeing the beneficial effect. But like it's always when you challenge something that's already published, the associate editor and the reviewers just said, well, it's not novel. There is no mechanism. It's negative data. We don't want that. I've talked about the triglycerides. I've talked about cholesterol-rich atherogenic lipoproteins. Obviously, I want to then spend some time also on the last class of lipoproteins, which are the HDL particles, the good cholesterol. And these particles, they can pick up cholesterol in the peripheral, for example, from macrophages in an atherosclerotic plaque, transport it back to the liver, where then the cholesterol can be excreted via the bile. And what we noticed in our studies already when we were activating the brown fat, what we saw that at the same time the HDL in this mouse model was increased during the study, and that the HDL concentration was an independent predictor of the atherosclerotic burden. So there was something going on in these mice on top of the atherogenic cholesterol lowering. And what we then did is we asked them, what is happening to these HDL, and performed lipidomic analysis. And here you see the PC and the lyso-PCs, the rest is also in the paper. But you see that there's a very pronounced remodeling of these HDL particles upon cold exposure and also CL treatment in various models of hyperlipidemia and also wild-type mice. So this really suggested that despite these relatively small changes in HDL cholesterol, we're actually looking at different species of HDL particles here. And we also looked into humans, and then what we found there is that we had lean and obese probands that actually cold didn't change anything. And of course, this was a little bit disappointing. But then when we performed these lipidomics, what we found is that there was really no remodeling in the obese, which then in the lean was visible. And at the end of the day, we have this lyso-PC 18-1 and the PC34-1, which are in our hands very good biomarkers for brown fat activity. And I mean, this makes sense because we know obese people have less brown fat. So we'd actually expect that maybe their brown fat activity is reduced and also the remodeling of the plasma lipidome is less pronounced. We also studied the function then of these HDL particles. And this is a classical so-called Dan-Rader assay where you incubate macrophages with radioactive cholesterol. And then the HDL picks it up and brings it to the liver, where it then gets excreted as bile into the feces. And you see here CL in cold exposed mice. And then over time, the radioactivity is more excreted when the mice are in the cold. So we also then studied the turnover of HDL by double labeling these particles with a cholesterol label and a protein label, and then perform various turnover studies and let's say some mechanistic mouse models to really delineate what is the driving force behind this. And I just want to point out here this model. This is an adipocyte lipoprotein lipase knockout model. Here you see that the particle goes, the clearance is not really so different between these two mouse models. However, if you don't have lipolysis in the plasma, you accumulate the cholesterol at much higher levels. So what this means is the turnover of lipoproteins in the plasma stimulates also the turnover of HDL and the excretion through the bile. So how is this then integrated? You can put these tissues into context in a way that here VLDL and acylcarnitines transport lipids to the brown fat, and we've heard it can also go the other way around. Here then HDL can pick up excess cholesterol, which also works for atherosclerosis, and then brings it back to the liver where it gets excreted through the bile. And there's also some recent evidence that these bile acids then again can stimulate browning and stimulate brown fat. So it's really a cool closed circle of lipid flux upon cold exposure. So in the end, I just want to spend some time on what is now the human relevance. I saw Denis Blondin, I think, was in the audience. So unfortunately, I have not mentioned any of his studies, but I also want to say that he's a leader in this field, and I've only picked his competitors. I'm sorry for that, Denis. So there were 2 papers in JCI this May in 2020. And you know, if I was humble, which I sometimes I'm not, I would say like these were great contributions. I think at the end of the day, I'm delighted to see that what we've been studying in mice, now there's proof in humans. And what they actually found is that this beta-3 agonist receptor, myabragon, really improves glucose homeostasis. And the paper by the Cypress group then also showed that it increases insulin sensitivity, but also had a strong effect on HDL cholesterol. And it really matches the mouse data now. I think a major conclusion here was that there was not really an effect of weight loss. It's much more that metabolic health was improved. So I don't think we're still quite there yet really to treat obese people with brown fat. However, I think these data really show that brown fat does have relevance for cardiometabolic health in humans. And it's great to see that there's another paper from Paul Cohn's lab, which you can find on bioRxiv these days, where they looked into roughly 140,000 PET CTs from 50,000 patients, and then stratified them into do they have brown fat, don't they have brown fat? And at the end, it's very clear. You can see that when they had brown fat, they had lower type 2 diabetes, less hypertension, less coronary artery disease, less congestive heart failure, and also the plasma parameters. They had lower glucose, lower TGs, and I cut off the HDL, but they also have high HDL if they have brown fat. So I think really ultimately, this is a full circle, and I think there is strong evidence that brown fat does play a role in humans. And before I get to my take-home message, I just want to quickly acknowledge actually not so much the people that have done the work. I just want to say thank you to my lab. This is a very updated group picture from last week. I have to say thank you to Jörg, my old PhD supervisor. We haven't worked on a project in quite some time, but I still look back to the good old days where we're doing some pioneering work in the plasma lipid fields. I'm very grateful to Patrick and also our dear colleague Jimmy, may he rest in peace, for the great collaborations we had over the year, and Christian Weber, who is my current boss, who lets us pretty much do whatever we want to do. And of course, also we have some funding sources. Thank you very much. And at the end, I want to highlight, at least for those that are tuning in from Europe, there's something called the European Lipoprotein Club. It is a meeting that is going to take place this year with social distancing. We've been able to run it. It's taken place close to Munich. It's a great meeting, even though it's called Lipoprotein Club. We have pretty much opened up to all cardiometabolic diseases. We cover topics from adipose tissue, NASH, to lipids in general. And it's very interactive. It's a very small meeting, so pretty much all the juniors get to present a talk this year, which is great. And I would really encourage you, if you have something that would fit the topic, please come to this meeting. I'm in the committee, shoot me an email, and I'd be delighted to see you there. Now, this is my take-home message. Brown fat in humans is real. I think if we all adopted a thermogenic lifestyle, we'd be all a little bit healthier. And I think the take-home message here is then you got to really train your brown fat, you know, go out in the cold and do something about it. So with this, thank you very much for your attention. I'm delighted that the organizers gave me the opportunity to share some thoughts and concepts here today, and I'd be happy if you have some questions.
Thank you, Alex. What a wonderful talk. When you put Claudia and you together, so it becomes even better. Well, you can raise your hands, everyone, if you want to ask a question. Otherwise, I'm going to start asking my question first. Okay. So, Alex, I think the brown fat and cardiovascular connection is an important one and completely unexplored since those studies. I have a question. So when you think of brown fats and thermogenesis in the context of interorganal communication between adipose and liver, what would be the strategy to benefit, you know, how would we modulate this collaboration rather than singularly brown fat for the benefit of cardiovascular disease? So what would be the manipulation? I can't imagine enhancing brown fat alone will be sufficient now that I've appreciated that this is an interorgan collaboration.
Yes, I mean, that's the golden question, of course. Is there a one-size-fits-all drug? Can we target NASH, brown fat all at the same time? I think, you know, there's some natural therapeutics called exercise, healthy diets. And I mean, it sounds silly, it sounds boring, but that's pretty much how it is. Can we really use brown fat as a therapy? I don't think so. I actually had a slide that was saying that despite the fact that human brown fat is real and that we've learned a great deal, also thanks to Sören and Camilla, we've learned a great deal about human brown fat in the last couple of years. I think, to me personally, it's much more, it carries some secrets of biology that we might be able to use for other projects as well, and maybe also some new therapeutic targets which may play other role and can be easily targeted, let's say, in the liver. And you might be familiar with the NRF1 stuff we've been doing in the past couple of years. This is a great example because there we found something in the brown fat which we are now only understanding that it is as relevant in the brown fat as it is in the heart, as it is in the muscle. It's just more difficult to see. And with some of the fundamental understanding of brown fat, we might get a better understanding of overall metabolism. Does this really answer your question? What would be the way to target that? There are some new triglyceride lowering drugs, which are actually lipid metabolism based. Bempedoic acid, for example, is in clinical development, and I think also is already prescribed. That is really maybe not based on brown fat knowledge, but it's really a lipid drug that's based on research on lipid metabolism. And I have high hopes that maybe because of the peculiarities of brown fat, there might be similar things we might be developing in the future.
Yeah, so I was also thinking that if we understood how they talk to each other, these multiple organs in this thermogenesis mode, that might also be a way to manipulate the system.
Sure. I mean, we've heard a lot about batokines, new potentially relevant hormones coming out of the brown fat. I am not aware that there is one so far that has proven to have a significant impact. I think also if you think of FGF21, that's much more of a hepatokine than it is a batokine, despite the fact that it might have some effects on brown fat. I'm uncertain, but definitely what the research tells you here is that any stimulation of energy expenditure, and it could be brown fat, it could be exercise, you name it, It could even be, let's say, caloric restriction, because that would also lead to an increased cycling of nutrients in the body. That this has benefit on your cardiovascular system, because the increased lipolysis in the vascular system has a beneficial impact not only on cholesterol lowering, it also improves HDL function. And I think understanding this connection better might tell us something for our everyday life, how we can use energy expenditure to improve our cardiovascular health.
Thank you, Alex. So I'm going to let other people have a chance. So John Sudoan has a question, so I'll let her ask her question.
Thank you very much. And first of all, thank you very much, Dr. Alex. A really great talk. And it's really interesting because lack of food and the cold exposure, if we think about evolutionary, those are two important parameters for the adaptation mechanisms. And I'm sorry, my background is entomology, so I'm just think about the evolutionary. And in hibernating beetles, and I just think about, because it's interesting, the brown adipose tissue. Have you ever, have you any idea about any animals like invertebrate, like insects has brown adipose tissue? Like, have you any idea? And what do you think about the evolutionary system? If you think about, can they adapt? Because insects has, they can eat lots of carbohydrates and they don't get obese, right? So those are my questions.
I think you touched upon many important topics here. Brown fat is much more of a latest invention by evolution. I mean, real brown fat, if you like, you find only in mammals. You find some uncoupling proteins also in non-mammalian species. There are some thermogenesis processes, and I think the creatine kinase pathway might even be the most ancient one that we know so far in also lower organisms. Why did mammals develop brown fat? I mean, if you think about a mouse that is in the forest and has to escape a predator, usually exercise and muscle activity would keep the mouse warm, would stimulate thermogenesis, and would prevent or would help the mouse really to survive in a cold environment. Now imagine it's in the forest and it's being hunted by whatever bird or a predator. Standing still without any muscle movement, the mouse still needs some good thermogenesis to survive, and that might actually when brown fat was coined by the Lord or by Mother Nature, whoever invented it. So I think there is some evolutionary advantage which we kind of don't really understand. For us, it just doesn't make so much sense in terms of energy in, energy out. Why would there be such thing if there are other tissues which can also burn a lot of nutrients? But I think this non-shivering is really an important key aspect for the evolution of brown fat. And then lastly, what was the third part of your question?
I just wonder, like, is there any insects has, do you think, is there any brown adipose tissue? Because they have their storage lipid in the fat tissue, and this is like liver and also adipocytes. So it's like a mixture package. So that's interesting also.
Yeah,
but
I think if you classify, I don't think it would it'll live up to the classification of brown fat. But I'm sure that there is still something we can learn about the lower organisms, and even plants have uncoupling proteins. So it's really not something that only the brown fat can do, but yet, of course, brown fat has a very unique special biology, the mammalian one. And of course, it is possible that in the fat body of Drosophila, other lower insects, there are still a lot of secrets which we can use to understand mammalian biology in an evolutionary perspective better.
Okay, thank you so much.
I have a quick question. So Alex, so most of the models that you've looked at, atherosclerosis, are you primarily looking at prevention of acquiring plaques? And have you looked at, once the plaque forms, can you get resolution of the plaque with cold exposure?
Yeah. Cold exposure, we have also looked into that, and that was part of the, let's say, failed experiments where we had to realize that cold exposure in the mice increases food intake so much in these models, particularly that develop plaques. It's not a feasible thing to do. But what we did was actually do, again, turn to the beta-3 agonist and take LDL receptor knockout mice that have pre-existing atherosclerosis, dyslipidemia, and then switch them to away from the cholesterol diet. Let's say you go to the doctor and he says, you know what, change your diet, activate your brown fat, and then see what's going to happen. So when we did that, what we saw that the plaques didn't really improve in size, and we saw some remodeling, so they were a little bit more stable. The blood lipids went down, but I have to tell you, we never published, I mean, yes, we have published the first set of experiments actually. The major effect, what we saw was actually first happening in the liver. So the bottom line is, I think, the way we did it was not proper, and we've never really followed it up. But I think there's some perspective there that you have to, at least in a mouse, do it for like 3 months or so to really see a significant reduction in atherosclerosis. That would be my guess. So it's a relatively long run, and I guess if we did it in a more, let's say, human-like model, it might be faster. But we have not done that. So I think the take is, it takes as much to make the atherosclerosis go away as it does to develop it. And also the mice don't really get myocardial infarction. The plaques in mice are relatively stable. So what we would need to do is a destabilization model and see whether also brown fat activation would stabilize the plaque, not really necessarily changing the size of it.
Yeah, great, thanks.
One more question from Zoe.
Yeah, I have a quick question is, you know, I just wonder how much LDL receptor contribute to brown fat thermogenesis. And if you compare the wild type and LDL receptor knockout mice, just put them on top fat, not like cholesterol, you know, atherogenic diet. So do you say the LDL receptor knockout mice are less tolerant to cold?
They perfectly survive in the cold. The only problem they have is because they get such a massive hypercholesterolemia. If you put them in the cold and not use a pharmacological activation, they get actually cholesterol deposition in the kidney, and it's very nasty. They develop a kidney failure over time. That is why then the LDL, of course, does not survive the thermogenesis, the cold so well, but that is not related to thermogenesis itself. So if you measure energy expenditure, brown fat function in these mice, it's completely normal. They survive the cold perfectly fine.
Okay, thank you.
All right. I think we're good with questions. I'm going to say Alex needs a rest. So thank you so much, Alex and Claudia, for these great talks. I certainly learned a lot. And
thanks to the audience for coming. I hope you guys enjoyed this. Thank you very much. And thanks to the organizers for giving me the chance to present some of the ideas.
Oh, it was our pleasure. So we'll see you guys next week, same time. Have a good weekend.
Bye.